Gang. I'm Curator of Horticulture at the University of Melbourne's System Garden. In honour of Botanic Gardens, we are visiting Botanic Gardens across Victoria to, among other things, understand a little bit more about education, horticulture, conservation and community engagement. I hope this is an opportunity for people to learn a little bit more about Botanic Gardens that they didn't already know, and encourage them to go out into Botanic Gardens and appreciate all the value that they can provide.
Botanic Garden for me is obviously a great collection of plants, but beyond the individual plants and their scientific value is the notion of Botanic Gardens as very special social places as well. We have a number of groups who come to our gardens, or are very welcomed to our gardens, from high school students. We've got a beautiful local high school who come to our gardens to they study photography, so they're using our garden as a venue to come and find beautiful things to photograph. Basically, we're really trying to encourage children to look and be in a garden in a particular way. And what we try to do is role model the way we'd like people to be in the garden in terms of interacting gently with the space. But really we're trying to foster a love of plants and a connection to the garden.
The best thing about a botanic garden is that it holds all those collections of plants, that you can come here and visit the world without having to travel anywhere else. That you can come here and visit plants that you wouldn't see in your backyard, you wouldn't see in your local area. So we're in a process of expansion and building. So we've just created our Garden for the Future, which is under 12 months old. We've also got the oldest public garden in Bendigo as well. So our heritage garden is over 160 years old. So hopefully we'll be replacing some of the older trees in the gardens in the heritage section. We'll also be joining the new garden with the old, so there'll be a new transition zone between the two gardens.
I think the really great botanic gardens are botanic gardens that are hubs. They're social hubs, they're environmental hubs, they're conservation hubs, they're horticultural hubs. The role of botanic gardens in conservation, what we're doing with BGANZ Victoria is looking at establishing conservation collections in a number of regional botanic gardens across the state of Victoria. But for that project to be sort of driven and supported by the Royal Botanic Gardens Victoria. Essentially what we're doing is we're going out to regional botanic gardens, we're working with the gardens curators and gardens managers on an approach to establishing collections of rare and threatened plants that are significant to that particular area. And then the RBGV will actually propagate all that, collect, source and propagate all that material, and then distribute that material back out into regional gardens.
So a botanic garden, for me, means that it's a place for people to firstly enjoy, but then it has all those other benefits of labelled plants, documented collections of plants. The importance of botanic gardens to communities, I believe, is that it gives them a sense of pride, it gives them a sense of place, and we've had people come on guided tours here, and one particular comment said the Melton Botanic Garden has put Melton on the map. To a large extent, this garden is the community, because the people have been the ones pushing it, backing it. The feedback we get from people who are visiting, walking through, doing recreational activities here is just fantastic.
A community space.
It's a garden to display a collection or a variety of plants.
What is a botanical garden? The most beautiful place to come to.
It's a wonderful refuge for the community and for visitors.
I think the free access is the best thing.
For me personally, it's the joy that it brings to people.
Everything. I guess, we need plants for everything to live, really.
Education, conservation ideals and public enjoyment.
Knowledge and inspiration and understanding.
Well, I'd be biased and say the Amorphophallus.
Banksia. In Australia, red gum trees.
A plant with gum nuts in it.
It's the eucalyptus.
Eucalyptus.
Eucalyptus gum tree.
Botanic Gardens Day is the last Sunday in May, so put it in your diary.
Well, good evening, everyone, and welcome to our second BGANZ webinar. The topic for this evening is rare plants, large and small, and I am really excited that our BGANZ month webinar series is on again, and we've got another talk coming up next week, which I'll explain to you when we get going. Now, I'm Costa Georgiadis, for those of you that don't know who I am, and I'm a Botanic Gardens Australia New Zealand BGANZ ambassador. I'd like to welcome you to our second webinar. And before I get underway, I just want to let you know that I'm actually up here in Queensland, and I would like to acknowledge the traditional owners of the land where I'm broadcasting from, which is the Turrbal and Yagara peoples, their lands and their waters of the Brisbane area. And I acknowledge elders past, present and emerging. And while we're on that, I'll just throw it to each and every one of you to pop up into the chat. Whereabouts are you coming from? Which town? And of course, which country are you on? So we can all learn something from where you are streaming. Of course, for all of those of you that are here, you're here because you love plants. And Botanic Gardens Day, as you heard, is coming up at the end of the month. But we've got these 4 talk series that are going on, and we have 4 wonderful speakers joining us, or 3 actually, and the topic we are talking about is the large and the small of plant adaptation and rarity. So let me have a quick look at some of our comments coming through. We've got Lexi up in Woodville in New South Wales. We've got Amanda coming in from South Australia in Mount Pleasant. We've got the Blue Mountains, Darug and Gundungara land. Thanks, Phil Schmidt. Robbie Beals coming all the way from East Gippsland. Ange Oliver's on the Gold Coast. Maria, Teresa, Earth Australia in the Northern Rivers. Thanks, Ryan. Welcome, everyone. What a great spread. We've got already a fantastic audience from all over the country. Waratah in Tassie. Big welcome to you, Genevieve. And Gladstone in central Queensland. What a wonderful mix. We've got Hayfield, Tim Ubergang. We've got Gubby Gubby, Robin Matthews, Central Coast, Darkinjung land for Kat Sopper. So big welcome to all of you. And it's wonderful to have you all join us for our talk today. Our 3 speakers, we have Matt Coulter, Tobias Hayashi and Ryan O'Donnell. And I'd just like to take this opportunity to get them in. Here we go. Matt is joining us. Good evening, Matt.
Hey, Costa, how are you?
Lovely to see you. It's great to have the session underway. Also joining us is Tobias. Welcome, Tobias.
Thanks, Costa.
Great to have you here. And last but not least, we have Ryan. And there's Ryan. Good evening, Ryan.
G'day. Good to be here.
Great to have you here. Now, Matt, can you just give us a quick little intro on who you are, where you're working, what you do, and we'll just do a quick round robin before we start with Matt's presentation.
Thanks, Costa. I work for the Botanic Gardens of South Australia, where I'm a senior curator in the nursery. I've been doing that for the past 14 years. Previous to that, I've worked in private nurseries. So my passion and background is in plant propagation and production. And yeah, the job I'm doing at the moment, I just love it so much. It's a dream job, get to work with so many interesting plants. And hopefully I'll get to share one of the really interesting plants that I get to grow tonight.
Fantastic. Now, for anyone a little bit cluey on plants, they might notice Matt's background, and that's not a screensaver. That is the real deal. But we will get to that a little later. Ryan, give us a little bit of an intro on where you are, what you're doing, and sort of why you love plants.
G'day, so I'm Ryan. I'm currently a PhD student at the Research School of Biology at the Australian National University on Ngunnawal country in Canberra. So moved down here in January. So after I finished my master's last year, and I worked primarily on the taxonomy of mint bushes, actually. So yeah, Chrysanthera, if anyone's cluey with plants.
Oh yeah,
good fun. Yep, love a good mint bush. So.
Yeah.
Got a couple more in the pipeline there. A couple of new species coming soon, hopefully, fingers crossed. Yeah, so why I love plants. I was always a really sort of geeky kid, and I was, very, very interested in plants and nature and everything. And my, grandma spent all the time taking me to the Botanic Gardens and checking that out. But I sort of fell out with it across high school. We had a really dreadful science and maths department. And I became like the English music drama kid. And I was actually, before I was a scientist, I was actually an opera singer. So went to the conservatorium and did my training and worked in opera. But alongside that, I did an amateur production of Little Shop of Horrors. And I was like, I used to grow those as a kid. Maybe I'll do that again. So I bought, this hanging picture plant off a hobby gardener on Facebook. And I just became absolutely obsessed with it. It started with just how to grow it, how to look after it. And then I just started learning more about the whole family and its evolution and its ecology and its conservation. And after a while, I sort of realized that I cared more about plants and nature than I did about singing. So I decided to make that leap, and here I am chasing orchids.
Fantastic. Well, thanks for that little bit of background, because I might just throw that to the audience. If you've got a little moment that got you into plants, just throw it up in the chat. And now, Tobias, welcome to you. Tell us a little bit about where you are, what you're doing, and your connection to plants.
Thanks, Costa. Yeah, so I'm coming towards the back end of a PhD that I've been doing through the Australian National University as well. I'm actually coming to you from Elcho Island in northeast Arnhem Land on Wittunga country. So wow. a bit of a distance from the ANU. But yeah, so I've just submitted my PhD not that long ago, and I'm currently working as a conservation officer at the Department of Agriculture, Water and the Environment. But yeah, I've always been interested in things that move, birds and animals, and now things that don't, orchids, I guess. And just always interested in natural worlds. So.
Oh, fantastic. Well, thanks for joining us, all three of you. And now we'll head over to Matt, who's going to give us the first presentation for the evening. Matt, take it away. I'll just. Do you want to start up directly, or do you want the presentation straight up?
I can just have a bit of a chat to start off with Costa,
yeah, sure.
And then we can get into the presentation. Firstly, I'd just like to acknowledge that I live on the Adelaide Plains. It's the traditional custodians of the Kaurna people. I'd like to pay my respect there, past, present and emerging. What I'd like to talk to you tonight about is the corpse flower, the Amorphophallus titanum. It's a plant I've been curating for the last 14 years. And 14 years ago, we received 3 seeds. And when I first started growing this plant, I really didn't know much about it. But in the past 14 years, I've carried out quite a lot of research and talked to people from all over the world. And I'd like to go through the presentation of some of my work. I'll just try and open this presentation. No, can you open the presentation, Costa?
Yep, here we go.
Okay. Is the slideshow working, or?
Can you put it onto the big screen?
There we go. Well, it doesn't seem to be starting.
Okay. We'll just try it one more time. Otherwise, I'll pop it up and.
Yeah, you might have to get the slideshow 100% not opening.
Okay, no worries, everyone. I'll just go to plan B, and away we go.
Okay, so this is some of the plants that I've actually learned how to propagate, which I'll be talking to you about in a minute. So we've actually amassed probably one of the largest collections of the species anywhere in the world on one side. So from those 3 seeds which we started originally, I now have over more than 100 plants of this species. And the importance of this species is it's an endemic plant from the Sumatran rainforest, and on the IUCN list of endangered plant species, it's now listed as endangered. And that was in 2018. Prior to 2018, it had been listed as vulnerable, but in that time frame, the species have been declining as well. It's quite a remarkable plant. Can you go to the next slide? It's not. Anyway, I'll talk about this. So in 2013, I did some research to see whether we could propagate this vegetatively, because when you had those 3 seeds originally, I wanted to see whether we could propagate it. So there's been some work to talk about that you could propagate it by leaf cutting. So I set up this trial, which became very successful. And on your screen here, you can see that's a leaf cutting that actually developed a root system. Okay, the slides don't seem to be going through. Are you able to scroll through?
Yeah, you just tell me when and I'll change the slides for you.
Yep, can you go to the next slide? There's just some other forms of showing the root system. So we go to the next slide. So after some time, this plant will actually start to form tubers, and then the leaf will die. So this plant actually has a massive tuber. So our largest tuber so far is 75 kilograms, and the world record is 150 kilograms for a tuber. So it's quite massive, it's quite an incredible plant. Go to the next slide. So those seeds are received in 2006. We flowered the first one on the 29th of December. And what you see there is actually a typical plant for an aroid. So it has the skirt, which we call the spathe, and then the spadix coming up in the middle. The actual flowers are actually down in the middle, which we can't see. And that spathe is actually a modified leaf. Go to the next slide.
Matt, can you just come a little bit closer to the mic?
Yep.
Thank you.
Is that better?
Yeah.
Yep.
It's not. It won't echo as much.
Sure. So the first one we actually flowered in the Mount Lofty Botanic Gardens where I work, and then the second one we flowered in the Bicentennial Conservatory in the Adelaide Botanic Gardens. And quite a remarkable plant. People are really interested in this plant. So the first one, we had 5,000 people come and visit, standing in line for 2.5 to 3 hours to spend some time with the plant. And then the second one,
5,000, really?
Yep, it was 35 degrees as well. It was quite amazing. And then the next day, and then the next flower, we had 10,000 people. So at this stage, since that first flowering in 2005, we've actually flowered them 11 times, which is quite remarkable. We've had a flower most years now. It's quite a difficult plant to actually bring into flower. So our successes have been quite, been really pleased with what we've been able to do. So basically, it's called the corpse flower because when it flowers, it emits a smell of a dead animal to actually bring the insects to it to actually pollinate it. So basically, after 10 years of growing, or 10 to 15 years is when it will flower, and it only has one night that can get pollinated. If it doesn't get pollinated that one night, it could be another 3 to 5 years. So the female flowers are active on the first night, and then the second night, the pollen starts to be released, but they change it so it can't actually pollinate itself. It has to get pollen from a different flower to it. Go to the next slide. It all happens quite quickly. So it opens up the first day, and then by about a week later, the whole thing will collapse. And then if it gets pollinated, then the infrutescence will stay up for a whole year, and then the seeds will come from it. So this plant is actually classified as the plant with the largest inflorescence of any plant in the world. The world record in the Botanic Gardens is over 3.5 metres tall, which all happened in about 8 weeks. So basically, the life cycle of the plant is below the soil. So the tuber is below the soil, and then above the soil is all annual, so whether it be a leaf or a flower. And when we had our third flower, we actually had a leaf and a flower at the same time, which never happened anywhere else in the world before. Unfortunately, that tuber then did perish. It flowered 2 years in a row, which is actually never heard of as well. Go to the next slide. So this is what the actual flowers look like. So this is inside the spathe. So what you see, the yellow part there is actually the pollen, and you see over to the left-hand side, you can see the pollen is actually shedding. And then below that is the female flowers. And you can see some pollen is actually being deposited onto those female flowers, but they actually won't be pollinated because the second night, the female flowers stop becoming active. So it's quite incredible that you wait all these years for it to grow, and then it's only got one night that it actually has that ability to pollinate. And the interesting thing about the aroma is actually pulses. So it's not just one smell. Actually, the further you're away from the flower, the greater the smell is. And they're quite complex compounds. About 30 different compounds they've isolated to actually trick the insect to come, because the insect thinks it's going to lay its egg on the rotting carcass. And it's basically a thermogenic flower as well, so it heats up. So that spadix at night time actually smokes, all this steam comes off it. And if you actually touch it with your hand, you can actually feel it's actually hot, and it's actually the temperature of a body. So, and then the spathe has that dark colour as well to actually mimic a dead animal. So it's actually quite interesting. Next slide, thanks. So this is a close-up image of the female flowers. So basically this thing is classified as an inflorescence. So each one of those female flowers can actually make seed. And if you get a strong pollination, you can actually make up to 300 seeds. But unfortunately, as I said earlier, this plant is actually endangered and it's wild, and there's lots of deforestation, palm oil plantations, etc. that's actually happening. So yeah, it's in a lot of danger. So having an ex situ conservation, which we're actually doing, is actually really important for the survival of the species. Next slide. So what we do, so this plant the seeds can actually be stored for a long time, like a lot of seeds can be for endangered plants. So what we actually do is actually collect the pollen and store the pollen, and then at a later time, that pollen can actually then be involved in cross-pollinations. So there's a research project at the moment with the Botanic Gardens Conservation International and Chicago Botanic Gardens, where they're actually testing, doing DNA analysis of all Amorphophallus in the collections, and we've sent some of ours. We have 6 different genotypes, and so in the day, if we need to cross-pollinate these out in the wild, we need to get the greatest genetic diversity as possible. So it's almost like what they've been doing in zoos for many years, where they actually have stud books, and they actually look at the relationships between individuals. So all that information is being kept, so when they get to pollinate, we're actually going to be pollinating ones that are actually not related to each other. And this program is called TREES. It's Tools and Resources for Exceptional Endangered Species, and there's 6 different species that are involved, and one of the Amorphophallus titanum is one of them. So it's quite an amazing plant. We flowered it 11 times, and yeah, it's I've been working on it for 14 years. We've got a network of people in botanic gardens around the world that are involved in it, and we talk quite regularly. It's a really difficult plant to actually bring in flowers, so because we flowered it 11 times. So from those 3 seeds, we actually made 7 tubers, and we've flowered every one of those 7 tubers now. And the ones that we actually propagated in 2013 are coming to age of flowering. So one of my main aims or goals with the plants is to see if we can actually get natural pollination, which has never happened in cultivation previously. So it's quite an amazing plant to be involved with. I've learned so much from it, and yeah, it's just really fascinating. I think that might be the last slide.
Is that the last slide?
Oh no, here's some photos of the flowers. So you can see that really, this is some of the flowers that just flowered recently. And we've got some really great ones that have almost black spathe. And these have been some of the ones that have been the darkest ones that anyone's seen anywhere in the world. Our biggest one to date is about 2.5 to 2.6 metres. And yeah, it's anytime one flowers, it's quite a remarkable thing to be able to see. And yeah, we've actually flowered one in winter as well. They generally tend to flower in summer. And then when the flower dies down, then the leaf starts the whole thing going again. So yeah.
Wow, Matt, that's there's so many questions have come in. I've got a few racked up here that people have asked. So if you just want to fire off some answers. Beck asked, okay, here we go. Do bees avoid it? That was from Karen.
Yeah, bees don't go to it. So the main pollinators are sweat bees and carrion beetles. And that one night that it actually that it gets to pollinate, the whole space basically turns black with sweat bees all over it. So sweat bees more like a little flyer, but yeah, normal bees don't seem to go to it.
Tessa asked, is this leaf growing roots from the corm joint or from a cut in the middle of the petiole?
No, so the leaf grows straight out of the tuber. So for 10 years, and every year you get a new growth, it'll be the leaf getting bigger every year. So the other thing I didn't actually mention is actually it's classified as a single leaf, which can get to 5 metres tall. So when the flower dies, then the new leaf from that flower comes on the edge of where the peduncle was. So, and every year, and then when the leaf is coming out of the tube, it comes out of the middle, but if it has a flower, then the new shoot comes off the side of where the peduncle was.
Wow. Now, Karen Gerg asked, does it need a lot of fertiliser?
Needs lots of water. If more you fertilise it, the bigger the tuber will get, and then the likelihood of getting the flower. So we feed it regularly just to be able to show that we can show some to the public every year. So since that 2015, except for 1 year, we've had a flower every year. And some years we've had 3 flowers in the 1 year. And also there was one time we had 3 flowers in 1 month, and 2 flowers were only a week apart, which was close to that, getting that natural pollination, but we actually have to have it 2 days apart. And when we get that, that'll be a really exciting time for us.
Wow, it's an absolutely fascinating, all the comments here are just like, wow, incredible. Glenda said, love these plants. This is extraordinary. So on and so forth. People are blown away. We've got a question here. What's their biggest threat in the wild?
So deforestation for palm oil plantations, and also poaching. So there's another species called Amorphophallus konjac, which people may have heard of. It's a thing called konjac noodles, which is made from the different species, and a lot of people are actually digging this up and trying to sell it as Amorphophallus konjac as well. So, but the main threat is palm oil plantations. They're just basically bulldozing the forest, and then yeah, they're gone forever, because like it takes that 10 years to get them to the maturity, actually to set seed, and then yeah, if they're bulldozing mature species, then yeah, the likelihood of them surviving is quite remote.
Zorza said yes, deforestation, palm oil, exactly what you said. Emma Simpkins asked, can you predict when they'll flower?
Quite difficult, but usually by the size of the plant you can tell. Initially we all thought that it was the size of the tuber that would be that would actually tell, but some tubers, we've had some large tubers that have just made leaves, and others that have flower on smaller tubers. But once the emerging bud comes out of the tuber, once it's about 30 centimetres tall, then I can predict whether it's going to be a flower or not. And one of the most difficult things in predicting the flower is actually the day that it's going to open. Like always, people want to know what day is it going to open, what day is it going to open. But now for the last 3 flowers, I've actually predicted to the exact day, 2 or 3 weeks out, which is, because I've got such a wealth of photographs of the flowering events that we've had, I can actually get quite close to the actual day that will open. And the day that it opens basically opens almost like clockwork between 12 and 2 in the afternoon. And once it starts to open, you can actually physically see the spathe move. That's how fast it actually moves. And within about 3 or 4 hours from being totally shut to totally open. So it's actually watching one open, it's just quite a remarkable thing. We thought that people may not want to actually keep seeing them, but every time we show one, we get more people that come and want to view them.
Now, we'll just go with this as our last question for now, and if any more come through, we can ask them in the general wrap-up after the other 2 talks. But Matthew Nicholson asked, when were they first described and by who?
Yep. So it was an Italian botanist working in Sumatran, Eduardo Beccari, and he discovered it in 1878 and dug up some tubers and sent them to Florence and Botanic Gardens in Kew. But those tubers didn't survive, and they sent some seeds to Kew, and they were the first ones to flower then in the 1890s. And there's actually an Amorphophallus named after Beccari, Amorphophallus beccarii. But he was an Italian botanist working in Sumatra in 1878.
Wow. And Hannah said, will you be selling seeds anytime soon?
No, so we haven't actually produced any seeds yet because we haven't done any cross-pollination. It's a monotypic plant. So if you actually pollinate it, the likelihood of the tuber can die, but people have actually proven in cultivation that you can actually get it to survive. So all that pollen that we've stored, one day we'll be actually doing some cross-pollination to produce the seeds. But in ex situ conservation, it's easier to keep the plant alive than actually to maintain the seeds.
Fantastic. Well, look, we're in the bush. Matthews said this is amazing. Kalamunda Garden Festival, loving this, so interesting. Matt, it's been fantastic. And I mean, it's such a plant. It's such an incredible plant. And for everyone to see. Did either of you, Tobias or Ryan, did you have any questions before we head into your presentations for Matt?
Not really, other than I'd love to see one.
Yeah, I've been dying to see one of these in the flesh. Like ever since I saw the photo of it, I'm just like, I need to see one of these. They look amazing.
Yeah, I mean, most people want to come and smell the plant, but there's so many interesting stories. The other thing I didn't actually mention was the actual leaf produces the thing which we call lichen mimicry. They actually produce spots on the leaves that actually look like lichen, so the animal in the wild actually doesn't think it's a tree. So even that in itself is quite remarkable.
That's a pleasure, Dan Austin. A great watch to drop into. I'm glad you managed to drop in. That's fantastic. So, Ryan, if you're ready, happy to throw over to you and share with us your tribe of Australian orchids and your presentation. I'll just get your Prezi up.
How's the volume? Can everyone hear me all right? Is that all good?
Yeah, your volume's good. Just while we're there, are you getting white noise from me? Because it is absolutely pouring outside and I can hear it on the roof, but are you getting any squelch from me or is it okay?
No.
Okay.
Sounds good to me.
All right. Well, everybody, here we go with Ryan O'Donnell's presentation. Thanks, Ryan.
G'day. So my name's Ryan O'Donnell and I'm a plant and fungal systematics PhD student at the Australian National University. So my research aims to order, classify and interpret the diversity of life on Earth. So the questions that I ask aim to figure out how and why organisms have evolved to be the way that they are and what forces are driving their evolution. And a good way that we can study these evolutionary forces is through one of the most diverse groups of plants on the planet, the orchids. So Australia has a lot of orchids and everyone loves orchids and it's not hard to see why. They are found on every continent except Antarctica and they inhabit a wide variety of ecosystems, and they've evolved to exploit whatever environmental niche you can think of, which is part of the reason for their success. So worldwide, there are approximately 28,000 species of orchid, and in Australia, we've got almost 1,700 of those species, many of which occur only here in Australia, and many of which feature pretty prominently on some endangered species lists. So if you see orchids in the wild, don't pick them. In fact, in Canberra alone, where I'm broadcasting from, we've got more orchid species here than in the entire United Kingdom. So of these 1,700 species, most of them can be split up between two major tribes. The first of which is the Chronichidae, which encompasses about 319 species, including the well-known Pterostylis or greenhood orchids, and their dainty cousins, the Spiranthes or the lady tresses. And sister to the Chronichidae are the Diuridae, which include about 1,010 different species, which represent the vast majority of the Australian orchid flora. So I'm sure many of our bushwalking viewers will see some familiar faces in this slide. We've got Diurus, Calodenia, Thelomitra, Calichylis. You know, the list goes on. But as you can see, they're ridiculously diverse. As I said earlier, orchids are extremely good at exploiting ecological niches, and one that they have managed to exploit so successfully are insect pollinators. So within the Diuridae, we can see the usual friendly flowers that give insects a nice nectar reward, but over millions of years, they've also evolved a few other tricks to get insects to do their dirty work for them. So these orchids will lie, cheat, and steal to get ahead. And some of them will look and smell a bit like food, and they'll trick their pollinators with a false promise of a free meal. But at the more extreme end of the spectrum, we've got these orchids that have evolved to look, smell, and feel like a sexy, seductive female wasp. So males in search of a mate will stumble upon these flowers thinking that they've found a rather willing lover, but it's all a lie. So try as they might, this wasp isn't going home with them, and they'll leave with their sexual energy spent with nothing to show for it except a great big load of pollen on their back. And if the fact that these flowers just exist isn't wild enough, if we look at this family tree of the Diuridae and the Pterostylis there on the side as an outgroup, of which many species themselves are sexually deceptive, we can see that these fornicating flowers have evolved several times, and it's not like they're all descended from a single sexually deceptive ancestor. This solution has arisen multiple times independently, and for some reason, Australia seems to be an absolute hotbed for this sort of deception. So to talk about this in more detail, I'm going to pass on to Tobias, who'll talk about this in more explicit detail, except in the Pterostylis orchids.
Now, Ryan, before we go any further, Carol Marshall has just said that she's seeing that house on the hill in Canberra is perhaps a big greenhouse full of liars and cheaters. That was a very sharp comment there, Carol, given what's going on in town at the moment. Wow, there's some great comments coming in. Tim asked just before we head over, how did they evolve to deceive?
We don't really know in a lot of these cases. We don't really know why Australia in particular harbours so many of these, or we don't know what particular factors drive the evolution of that system. It might be a high proportion of a certain pollinator that's available that can be exploited, or the type of insects that tend to be of the bees and wasps that tend to be exploited. They tend to be solitary species, so they're not the ones that live in great big colonies. They tend to be on their own, and so they rely more so on detecting pheromones in the air and letting being able to sniff out those potential mates. So we don't exactly know how that has happened yet, and you know what has driven that, but it has become a very successful system. So it happened multiple times, and like it doesn't occur just here in Australia. It occurs. There are a few in the UK, throughout some in Europe. There are some who say that this sort of system occurs in South America, but I think it hasn't explicitly been demonstrated yet. But yeah, I think Tobias will have some much more detailed answers for these.
Yeah, all right, because there was one question here. So the insects and orchids co-evolved.
In a sense, yes. In some scenarios, these orchids are very closely, tied to their particular pollinator, and they'll be, oftentimes it's like a 1-to-1 ratio, or it might be not exactly, but they've evolved to mimic a very particular kind of wasp, and they'll differ in their appearance and their chemistry even.
Fantastic. All right. Well, there's plenty of questions coming in. Let's roll on, Tobias, and carry on from where Ryan left off, and then we can get back to all the questions. So, to everyone watching, keep racking up your questions for Matt, Tobias, and Ryan, and we'll go through them when Tobias has gone through his chat. So, Tobias, will we start with the slides?
Yeah, start with the slides. Thanks, Costa, and thanks, Ryan, for that nice introduction to the world of sexual deception. So in my PhD, I focused on pollination in the genus Pterostylis, or the greenhood orchids. And I'm sure many of you will be familiar with at least some of these. And pollination Pterostylis has received somewhat less attention than some other orchid groups, such as the spider orchids, or bird orchids, or hammer orchids. So it was my mission to uncover some of the secrets of greenhood pollination. Given the short time for this presentation, I'm just going to focus on one of my study species, but I did quite a few, worked on quite a few others throughout my PhD. And the one I'm going to focus on now is the swan greenhood, which is pollinated by males of a single species of fungus gnat. Now, fungus gnat is a little fly that you might have noticed in your greenhouse, perhaps. These fungus gnats that are involved in the pollination of this orchid are not pests. So this swan greenhood is a small orchid found in grassy woodland in southeastern Australia. And it has small pale green flowers with a prominent dark appendage on the labellum. If I just switch to the next slide. So you can see they've got quite small flowers and a prominent appendage on the labellum. Now, the labellum is a modified petal and is a characteristic feature of orchids in general. But in Pterostylis species, the labellum sits on a hinge and is triggered by movement. And you can see the right-hand side set of photos. I've cut the side of the hood away so you can see inside the hood, or the galea. And in the top photo, the labellum is in its set position, and in the bottom photo, the labellum is triggered shut, which kind of forms a bit of a trap system with the hood. So let me play you a couple of videos to demonstrate how this works. If you could cue the videos, Costa. Here we go. So you can see a male Bradisia fungus gnat on the flowers from the family Sciaridae. It approaches the labellum appendage. Whoop.
Oh.
And away it goes. So this is an undescribed species of fungus gnat, like many Australian fungus gnats, and you can see it waves its wings while it's on the orchid. Then it spots the labellum appendage and it attempts to copulate with the labellum appendage. And this causes the labellum to trigger shut, and the gnat becomes trapped inside the flower. If you just want to go back to the slides there, Costa, I've got a bit of a photo-by-photo replay, if you like. And so the gnat becomes trapped inside the flower, and then once it's trapped inside the flower, it is forced to escape by a tight passageway. and it collects pollen on its back on its way out of the orchid, as you can see in the last photo there. So how does the orchid trick the male gnat into copulating with it? Well, it first attracts the gnat by a scent. So the scent that the orchid produces is probably the same, we suspect it's the same as the scent produced by the female fungus gnat. And of course, this makes the males very interested. And because sex pheromones are so specific, this scent, which attracts the male gnat, only attracts one species of male gnat. And because the attraction is due to scent, if you hide the flowers, the male gnats are still attracted, provided the scent can disperse, of course. However, in this case, scent isn't everything. So the scent which attracts the male gnats is produced in the flower, but we know it's not produced in the labellum and in the labellum appendage. So if you hide the labellum appendage, the male gnats are still equally attracted to the flowers, but they don't attempt copulation, which suggests that the labellum appendage might be acting as a female dummy to kind of confuse the gnats, I guess. So in summary, both good smell and good looks are probably needed for the pollination of the swan greenhood.
Wow. Wow. That is amazing, Tobias. And together with you, Ryan, like the people are, Tessa said, poor confused gnats. Good grief, an orchid that kidnaps and imprisons. Like, this is, it's so wonderful to see it beautifully presented and articulated like that, so people can realize that when they're walking in the bush, we have these incredible plant processes taking place. And, from Matt, from yours, this massive thing, this 3 meters tall to what Tobias and Ryan are talking about, cutting it open and being this tiny little stage, but so much is going on.
Yeah, incredible. It's pretty crazy. It's pretty crazy with with these sexually deceptive orchids, like the orchid has to work out how to produce an insect sex pheromone. How does that happen? What are the odds of the orchid suddenly working out how to produce a sex pheromone from a completely different kingdom of living creatures? It's crazy.
Rebecca has just said good looks and good smells like me. Well, I don't know about that. Someone asked if we could see the video again, and look, I'm going to make a sort of an executive decision on this one and say that video is too good not to see again, particularly for anyone that might have just dropped in. So oh no, that's the slide. Here we go. Let's have a look, and feel free to walk us through it again, Tobias.
Yeah, so it's really the it's the scent which gets the gnats to the orchid, but when they're on the orchid, this little black appendage, we think, is kind of like the rear end of a female gnat, perhaps, and so the male really hones in on it and then grabs onto it, and then next minute trapped.
Incredible footage, really incredible footage. We've got some great questions coming in here. Having the labellum remain, I'll just throw this one up on the screen. Having the labellum remain retracted on a plant with a single flower makes sense to prevent self-pollination, but how does it aid in a species with multiple flowers?
Yeah, it's an interesting question. So in these systems, the labellum kind of functions to trap the gnats. So if the labellum isn't triggering for whatever reason, then the gnats won't pick up pollen because the pollen's kind of hidden in this passageway, and they wouldn't ordinarily go through the passageway if they weren't kind of forced to. So I guess in all Pterostylis, in the single flowered ones and in the multiple flowered ones, the labellum is kind of required to close off, the trap so that the gnat is forced to escape via the passageway and therefore pick up pollen.
There was one more question, and then I'll go open to general questions for the three of you. The whole, where was it? How many hours did you have to wait to get footage like that? That's a good question, Emma Simpkins.
Yeah, this was research that I did over 2 field seasons, probably about 2 or 3 weeks in the field for each season, so 5 or 6 weeks. But yeah, it's a question of finding a good population of gnats. You can find a good population of gnats that are going to respond to flowers, then you can get some nice footage pretty quickly.
Joe Ford reckons it's another good reason I'm glad I'm not a bug. Tessa asked, would this change the mating dynamic of the gnats with male gnats becoming leery, or are there so many gnats that the orchids are barely a blip?
Yeah, that's a great question. We know in the wasp pollinated systems that the wasps actually learn where the orchid is and they avoid it. So they learn the location of the orchid and then they don't visit again. But we think with the gnats, they probably don't learn, and so they probably just kind of keep on visiting every now and then. But it's probably not a big cost to the gnat, because kind of securing a mate is such an important thing, and they'll kind of respond to as many, whatever smells good, I guess. So we don't think it's a real big cost to the gnat, but it certainly takes time out of their day, so to speak.
Nadia said this is the best live show for wow, wow, and big wow. So thanks to the three of you. A question here for you, Matt. Are there Australian relatives of the corpse flowers?
Yep, so there are 2 species that occur in Australia. There's Amorphophallus pinifolius. It's not endemic. It occurs in northern Australia, but it goes up to Papua New Guinea, up into India. And then there's another species, Amorphophallus galbra, which occurs in Northern Territory, Western Australia, and Queensland as well. So they're the only 2 species that do occur in Australia.
Now, Zorza has said Ryan's jumper, catfishing orchids, and rotten corpses. Best talk and footage ever. So you guys have definitely captured the crowd here. Now we've got some more questions. More questions coming in here. I'll just okay. Carol Marshall asked, and this is a good question. I wonder what the system is that enables movement of the labellum appendage. Do plants have muscles?
Yeah, this is a great question, and it's something that I wish I knew the answer to for Pterostylis. I think there's a PhD in there for someone who's interested in that. Yeah, it would be really interesting to know what mechanisms kind of trigger. There's some interesting research on the Venus flytrap to do with the shape of the leaves kind of being a certain way, and then when they flip, the shape changes, and that enables the shape to kind of the leaves to move really quickly in a way. But yeah, I would love to know the answer to that.
Yeah,
in a lot of those sort of moving plant systems, a lot of it comes down to sort of chemical and water changes that are triggered by the touching of certain hairs or something, and so basically there'll be a change in chemical composition, and that will cause a change in pressure sometimes, and it'll make the leaves close or move. But yeah, I'm not sure if it's been studied in Pterostylis, but yeah, there are no muscles, so to speak.
We've got a question here from Glenda. How long does it take roughly for the wasp to escape via the passage?
For the gnat, yeah, that's a good question. It really depends. Some of them go through in a minute or a couple of minutes. Some of them can take 15, 20 minutes, half an hour. sometimes they get stuck, particularly if they're already carrying lots of pollen and they try to go through again, then they can get stuck. And so yeah, sometimes you find dead gnats inside flowers, but they don't usually die. It's just it's an occasional occupational hazard of being a pollinator of these guys.
Yeah. An occupational hazard. It's kind of, did they get that the training manual on at birth? That's the question.
Yeah, it's an unwilling job, I think.
It definitely sounds that way. Phil Schmidt said that labellum movement is almost as good as an ultricularia.
Oh, yep. Nice. It's that's the carnivorous bladderwort plants.
Yeah.
Matt, David Odell says, I've seen a corpse flower at Huskerson. I was blown away when I saw it. I didn't think it was possible. That's down on the New South Wales.
Yeah, it'd be unlikely to be the Titan arrow. It's probably more likely the Dracunculus vulgaris, the voodoo lily. Because they're real, I mean, even in Adelaide, we can't grow them outdoors. They'd only survive outdoors in the tropics. But the Dracunculus vulgaris is a very strong smelling species as well that's in the same plant family.
John just said the juxtaposition between the largest flower in the world and the diminutive orchids tonight has been really mind-blowing. And it's wonderful that the three of you have been able to contribute your experience and have that contrast of scale and then intricacies in both of them, because they have such incredible mechanisms. I mean, the heat that you were talking about, Matt, I went and saw one out at the big succulent nursery in Melbourne, and they had one and they'd cut it open.
Yeah.
Do you know where I mean?
Yeah, I know the one you mean. Yeah. They called it Tiny Titan. Yeah, it was a small one. Yeah, Garden World.
Garden World, that's right. Yeah. And they had a thermometer there, and you could and you could feel the heat, see the heat, and of course get the smell.
People have done thermographic videos of it. You can actually see that as you go up in the spadix, the heat changes, because basically the heat gets drawn up the spadix out into the forest. So yeah, and sometimes you can actually see steam coming off the spadix. So when you see that, it's quite remarkable.
Yeah, Tessa and Zorza just said, yeah, Collector's Corner at Garden World. That's the one. That's the one. We had a question here, Tobias. What are you working on now in the NT?
I'm actually pretending to be in Canberra. I'm working for the Department of Agriculture, Water and the Environment as a threatened species assessment officer. So, yeah, just pretending like I'm in Canberra, but with 30 degrees outside.
Yeah, she's a bit cool in Canberra at the moment. Well, I'll throw that question to you too, Ryan. What are you specifically working on?
So I'm just at the beginning of my PhD. So at the moment, I'll probably also be looking at Pterostylis, looking at sort of species level questions and delimiting boundaries between some species which are quite closely related. So we'll probably be looking at some population genetic stuff and things like chemistry and gene flow and flower structure and morphology to try and quantify that difference and see how, if they are different species, and also how those species have probably formed, what's driving speciation between them. But then also, all orchids are what are called obligate symbionts, in that they rely on a fungal partner to actually germinate from seed. And so most of the fungi that these orchids partner with are undescribed. We don't know what they are. Well, we might know what they are at a genus level. We don't know what species they are. They just haven't been described. So that's particularly so in Pterostylis. There are at least 5 or 6 undescribed species there that associate with the groups across the this very large genus at the moment. And so part of my PhD will be looking at the fungal partners of Pterostylus and trying to describe and delimit some of those species as well, and understand what's driving that sort of evolution as well. Because the fungus that partners with Pterostylus in particular is they're microscopic soil fungi by the genus name Ceratobasidium, and it's very closely related to very severe plant pathogens by the name of Rhizoctonia, which are responsible for billions of dollars worth of crop loss annually across the world. So there's a lot to be gained from understanding how this fungi has, made that shift from being pathogenic to, being in a partnership. So yeah, trying to understand that relationship a bit better as well.
Faye has just asked this one. We have many Pterostylus orchids in WA. Will they all be pollinated by fungus gnats? I'll have to get checking.
Yeah. That's go out and have a look, and if you wait long enough, you'll probably see some. Yeah. So nearly all Pterostylus species are pollinated by fungus gnats. Probably most of them are sexually deceptive, so they pretend to be, a female fungus gnat. Some of them are not. There's one group of Pterostylus that don't occur in WA that are only on the east coast that are pollinated by other flies, but yeah, mostly fungus gnats. Yeah.
Thanks both for sharing what you're both up to. I'll throw that question to you now, Matt. What projects are you looking down the barrel of at the moment?
We're trying to grow the Amorphophallus species collection. So there's more than 240 species of Amorphophallus worldwide, and currently we've probably got about 15 species, and we flower probably about 10 different species. So yeah, looking at that, working on that, yeah, that's probably the area that we're sort of looking at to develop more, because we've got quite a big collection of one species, the titanum, but we're trying to get greater collection. Really, one day I'd like to do a hand pollination of one of the flowers, so we could actually show the public what the fruit actually looks like. Because if you get a fruit, it'll stay out for a whole year. Yeah, and sort of looking at taking some of the pollinator stories, doing some hand pollinations, and in the future, if we can actually get some natural pollination happening, would be really exciting if we could do that. So, as I said previously, the 100 or so plants that we've got, they're coming to an age of flowering shortly. They won't all flower at the same time, but yeah, to get multiple flowers in 1 year is really something that we're really excited about. And showing it to the public is just a wonderful thing, because there's so many stories. People come to see the corpse flower because of the smell, but you tell them about all the other stories about the plant, and it's just, there's so many remarkable stories about how fast it grows. When in vegetative state, it can grow anywhere from 0.5 metre to 1 metre in 1 week. The size of the tuber, yeah, so many fascinating things. And I'm really fascinated by the leaf as well as the flower. So at present, in the Adelaide Botanic Garden, we've got a collection of 22 plants on permanent display, and we've done a whole lot of new interp on it as well. Yeah, and it's just, I'm really excited to show the public there's more than just the flower, because there's so many other interesting things about the plants as well.
Wow, there's still plenty of comments coming in, and I like this from Faye about the gnats, and I knew insects are so important and deserve more respect. Thank you. I totally agree with you there, and I know that Ryan and Tobias are well and truly connected to the importance of insects. People are just passing on their thanks to the three of you. Thank you, Leah. Thank you, Barbara. And we'll take this as the final question. What are the most beautiful species of orchids which can be found in the tropical rainforests of far north Queensland? Is that within your realm?
Probably all something called Dendrobium, I would wager. Although, depending on your taxonomic flavour, they may not be called dendrobium. I think there have been several attempts to reclassify that whole group of orchids into something a bit more manageable than just this enormous genus that is dendrobium. Yeah, I think Mark Clements and some other orchidologists are still sort of working on that and pushing for the changes to go through.
Phil Schmidt has got a bit of a drop moment, Mike, just there, if you have a read of that comment. The terror stylists.
That's a good one.
That is. Yeah, you'll have to have a look at the comments when we wrap up, because there's lots and lots of appreciation. So informative. Thank you for sharing your knowledge, both moved and amused. Fantastic hour of learning here. Thank you all so very much. Super session. Thanks, Matt, Ryan, Tobias. Now, before we wrap, actually, there's a few more. There's plenty more thanks coming in. Thank you, everyone. Relief, Lake Macquarie, Robin Matthews. It's been fantastic, and as everyone has probably read in the posts, our series of panel discussions is all part of Botanic Gardens month, and we're working towards Botanic Gardens Day at the end of the month. So to all of you listening, after being inspired by Matt, Tobias and Ryan, find your inner botanist and take the Plant Lovers Challenge. There's some fantastic videos starting to get put up. I've shared, I shared one last night or yesterday on my Facebook page, one of the botanists, Beck, down at the Australian National Botanic Gardens in Canberra, and she shared with us her Plant Lovers Challenge. You can do it just as a video, or you can take photos and write a post. But it's really just about making this a month of awareness and of sharing, wonderful sharing from specialists such as we heard from last week, and of course this week, and heading into next week, where we will have next week's presentation is on, where is it? I've got it here somewhere. Of course, it's just disappeared. I think it's on plant hunting. Yes. So that's going to be another fantastic chat. There's still a few more questions, but look, I'll wrap it up there because we've reached the hour mark. Marie said it's great to see specialists coming together with a common passion. Thank you so much. What a wonderful webinar. Thank you for this amazing experience with such great visuals and information. So hope you're toting your gumboots. Take care. Have my thanks. I just made it home in time to watch this. Oh, that's a pleasure, David. So, wow. Thanks again to the three of you for taking the time to prepare and make the presentation on our second webinar. It's been an absolutely fantastic evening. I've got one last couple of slides to add here. This is our thanks for BGANZ, the next and to the sponsors that you can see here. I'll just get that that banner off from blocking the slide. Where is it? Find it about this. There we go. Plant Health Australia Foundation, Botanic Gardens, Seasol and Augusta. Thank you to the sponsors with BGANZ, and I've just lost that. That slide's gone a funny colour. Let me go back to that. There we go. Yes, so the 19th of May is plant hunting, and then on the 28th of May we've got paleontology and Gondwana, and that will be another two wonderful talks coming up. Same time, 7 p.m. Thursday next week and the week after. Ryan, Tobias, and Matthew, thank you all once again for a lovely, a really fascinating conversation. And if you have any final questions, drop them in the chat, and our guests will jump in there and have a look at those answers. So, from all of us here at well, what am I talking about? From where we are, I'll just take this. I'll take this out. Have a lovely evening, everyone, and we'll see you here again, same time, 7 o'clock next week. Ciao for now. Thanks, Matt.
Hello.
Thanks, Ryan.
Thanks
see
so
you,
much.
Tobias.
Thanks, Russell.